Hello, my name is Stephen, and here is a brief intro to an interview slash animation slash podcast with the one and only Dr. Pierre Lorenzo Puri, who is an MD slash PhD professor at the Sanford Brennan Priorities Medical Discovery Institute. He's part of two different programs at SBP. He's part of the Development, Aging, and Regeneration program. He's also part of the Neuroscience and Aging Research Center. A little bit by hold before we get too far in depth with what the interview is going to entail. He's a full professor at Sanford Brennan Priorities. He did his MD and his PhD, both in internal medicine at University of Rome. Here, La Sapienza. And he did his postdoc at UC San Diego in the Department of Biology. Just as a little bit of background for him. Now, what do you expect from this interview here? Well, glad you asked. In the interview, we'll be discovering here what it takes to get from becoming a doctor into a scientist. How does one transition from wanting to work with kids and patients to wanting to discover a cure or a potential cure for Duchenne muscular dystrophy? More exciting parts here, and sorry, UCLA students. Why UC San Diego? It's a subtle detail, but that's something you can find out somewhere in the interview there. After explaining these introductions, we'll go take a step back into a more traditional interview, looking at Dr. Lorenzo Puri's research here on HDAC inhibitors. You'll see this diagram come up again. Listen carefully, you might get some insight on what this paper is about. We'll also take a pretty ambitious overlap here between these three fields that combine together into a rather emerging one, combining statistics, biology, computer science. And seeing that the future of medicine and biology and the like comes not just from the wet lab, but from the dry labs as well. As we mentioned, the importance going into bioinformatics. And we finish up the interview here with a message for those who want to get into research, or for those that are thinking about becoming a scientist in the future. We'll look at 3, maybe 4 levels of inspiration, or levels that we need to achieve here in order to consider ourselves a scientist, and in order for us to do even more and develop new innovative techniques that can help us find a cure in the future, or at least get us one step closer to one. Okay. How do we develop new ideas? What is research about? And how do we inspire people and others to do the same? So join me on this wonderful, relatively long interview with Dr. Lorenzo Puri to find out.
Okay, cool. So the questions I have are not as technical, but more on how did you interested in DMD research, how do you address the community, and later on I'll ask a couple of questions about papers on it. And the project I want to try to work on is kind of making a little animation, kind of like what you'll see in Minute Physics or Minute Earth, in which they draw out a simplified version of how the science works. I think it's nice and simple, it gets the idea across, and it's pretty engaging as well, since you have to see moving parts instead of just still images on a PowerPoint. Okay, So the first question I have here is, how did you get started in research?
Interesting question. I like it. I was a medical doctor. I was working in Rome. I got my MD degree, and I was in my residency in internal medicine. And at that time, internal medicine, the part that I really liked was the action. So I was in the emergency room, spending a lot of nights in the emergency room. And I guess probably in one hand, probably I was too young, and I was a little bit burned by, or shocked, or kind of traumatized by a few things that probably you don't want to see when you're 24, that in the emergency rooms, unfortunately, you get to see. And at the same time, I started to see something that I didn't like in that job. I mean, there was clearly one part of the patients, probably the large majority of the patients, where you really understand that you can do something. You can cure them, you can treat them. But then I saw that there was another part of the patients where as soon as the diagnosis was there, you knew there was nothing to do. And I started to say, you know what? Is it more interesting to apply something that has been already discovered and we know is working? It is more rewarding, there is no doubt. And anyway, this is what doctors should do. Or it is going to be funnier to discover something, funnier, more useful to discover something to cure incurable disease. And I started to be interested in the second question. And the answer was in the research. So there was a laboratory of molecular biology in the same building where I was working, literally in the same clinic. And I started to get interested in research, and my target was were pediatric disorders, because I found unbearable to see, young children affected by disease, parents dealing with the disease and having as only answer, there's nothing to do. That's the last answer you want to receive from a doctor. And I said, okay. I started to work, in parallel, doing my work as a doctor until 4 o'clock. And then I was going down in these laboratories and start to do my own experiments. And it was progressive, these kind of things, progressively. I didn't even notice I was getting more and more into research. I literally fell in love with research. And then even when I started to understand that it would have been interesting to go around to do experiments. So I spent some time in Germany, in Berlin, because there was a technique that I found very useful for my experiments. It's called microinjection, where you can inject the DNA in the nucleus of the cells. In my case, they were muscle cells. And I came across an interesting finding. So one summer, I decided to go to United States, and I gave probably 15 interviews, something like that, until I got an interview at the UCSD. And I have to confess, more than the topic, I like the place. And I say, San Diego sounds a nice place where to do the postdoc. I like to surf. So I said, okay, so I kept on hold my position in Italy. And I said, I'm going to spend 2 years only research. But it came out that after 25 years, I'm still doing only research. So now my patients are those patients that I work for in terms of research. And I'm very interested in participating in clinical trials. So one of the discoveries in my lab, which is the potential of the pharmacological potential of HDAC inhibitors, is currently into clinical trial, and I help them to go into preclinical studies, early phase clinical trial, and now they're fully in clinical trial. So that's how I decided to, that's how I became a scientist. You know, after the postdoc, I liked so much to do this job that I got an offer as a PI, and I continue to do research, and I'm still in love with this job. This is a beautiful job, has an incredible potential, and I always say intellectual freedom. It's a lot of intellectual freedom.
Yeah, I think I'm on a very similar path. Like when I entered college, I thought I would just become a med school student, go into things like cardiology, something like that. Like that had that path. And then as soon as I used to see, like, because we're so research heavy, I got into research, and so I realized, like, this is something I'd rather do. Just invent new things, discover something new, rather than using a technique and then like study, and then use it, and then
that's exactly
that. Like it's also
that was my point. Yeah. I say, you know what, everybody can cure these guys because there's a treatment already, but how about these guys? And I was also, you know, it was a mental challenge, but also an empathy. You know, these guys are in the hospital. They believe that we will cure them. They don't even know. They trust us, but we don't have any tool. So I want to find the tool to help them. And that's exactly the point. You got it.
Yeah, actually, it's one of the things I'm glad about being here. It's all about research. We do have a really great med school program as well, but research is also important. Okay, so I guess like the next question here is, what got you into DMD in particular? What happened along the way to make you interested in that particular subject?
I initially, when you go to the lab, you cannot really pick your topic first, because you do what people do in that lab. Although I didn't want to do what they were doing. So the lab where that I joined when I was a doctor was mostly into, you know, focus on hepatitis, viral hepatitis, B and C. So I started to train, you know, as a scientist working on hepatitis B and C. But they didn't want to do that. So I asked to actually resume a project in the lab that was abandoned. And it was more on a different topic. It was on the effect of circulating factor called angiotensin II, which is implicated in cardiac hypertrophy and hypertension. And I wanted to study the hypertensive effect. So I really needed to study the smooth muscle cells, because those are the cells within the vessels that, after receiving signal from angiotensin II, contract. And the vasoconstriction is one of the mechanisms by which hypertension develops. And I got the wrong cells. So the PI on my lab gave to me the cells and said, those are smooth muscle cells. Okay. I started to study, and I say, they didn't match with the literature. I was reading the literature, I was looking at the cells, and nothing matched. So I went to a lab next, and I found out that the structure they were formed were more closely to the myotubes. That's what I read. So next to my lab, there was another institute where there was one guy who was working on skeletal muscles. I went over his lab and I say, listen, what do you think are these cells? Oh, those are the C2, C12. Those are mouse muscle cells, myoblast. Okay. So, you know, at that point, it was already some time I was working with these cells, and I started to work on muscle. And then at that time, it really came up the first evidence that there was this protein, this gene mutated called dystrophin that could have been the cause. And there were a lot of exciting experiments on in the field of skeletal myogenesis, mostly related to the discovery of the transcription factor that promote the myogenic program, MyoD, and several other proteins that behave like MyoD. So, you have to study something, and I got trapped by that. So I started to study MyoD, how it works. And I was really, really lucky because I had a couple of hypotheses, intuitions, if you will, that literally held true. So I was starting to get interested more in transcription. I said, how does a transcription factor works? And I postulated that transcription factors would have activated POL2, but I couldn't see anything that, based on what we knew at that time, that could mediate this functional interaction. So I postulated that a transcription factor that binds the DNA would have recruited some enzymatic activity. There was, it was a chemical process. So I don't want to get into the detail, but I identified that was one of probably the first identification of an acetyltransferase called P300 that was recruited by MyoD. So I was lucky to identify this interaction. And so at that point, I really wanted to get some money and start the research in Italy before I left for the United States. So I applied for this call. It was a call for grant on muscular dystrophy. And I said, let me understand what's going on there. And since that, I got the money. And originally I wanted to understand whether MyoD works properly also within the context of dystrophin deficient mice, which is still the topic, believe it or not, still the topic of my research, because research is endless. You make a discovery, and the discovery opens other 100 doors. And for each door you discover, the discovery is catalytic, the process of research is catalytic. Every discovery opens for new discoveries. So after that, I started, the no returning point was when I started to attend the conferences. Especially there is one type of conference where you see all the patients, all the parents, and this is a parent project, so it's an association of parents of dystrophic boys. And that was the no return, because before I was only engaged mentally and scientifically, but then after I saw the children, the parents, I got engaged also from another standpoint. It was a deeper motivation. It was more a human point of view. I really want right now still, more than ever, I really want to help these people. So that's the way it wasn't. If your question was, did you decide, at your age, I want to target this disease? No, everything happens because things, I think, happens in your life, and sometimes you got to let them know, let them happen. My philosophy is that whenever you start to plan, especially at your age, too much, you got stuck. Sometimes you have to let life, plan things for you and see, it's taking me to a nice place. Okay, it's giving me a good challenge. It's putting me in the right spot without planning too much. That's what I did.
That's pretty much my philosophy through college, is that the moment I start to plan things, I always just stay away. I deviate from the plan. So I just do whatever I want to do. And
that's smart. That's smart. Yeah. I like that. That's very smart.
Yeah. Because I feel like it's much more fun if you don't know where you're going and you let things happen. Then if you have a plan, you do X, Y, Z, and it's you've done the things you planned to do, but you didn't explore anything new. You just like you just focus on one particular thing, which I don't think it's
That's
bad.
right. Actually, you know what, Steve, this is the right mindset for a scientist. And I'm glad I can hear that sometimes, because most of the times I hear, I want to do this. That's my plan. That's my project. No, sometimes you gotta walk. You know, it's like when you land in a new city and you're thirsty, you have two options. You want, you let the agency to plan the whole travel, you know, all the itinerary for you. But there is nothing better than walk through the city and say, oh, look at this corner. I'm curious. I wanna know. I wanna understand. This is the attitude of the real scientist. Discoveries, and possibly not getting into what everybody think or what everybody do. You want to be original. The best quality for a scientist is the originality. Is to see things that others don't see, because you know, things that are seen by other people are taken care by them. You want to see it on a. It's a. I don't know if it ever happens to you. You know, sometimes you look for an object that you lost. If you look sitting from on the same position, I don't think you have a good chance. Sometimes you have to move around. You have to find your angle. And if this object, you know, it's in between many eyes and people, probably only the person that goes around and find an angle that nobody took will find the object.
Actually, I'm gonna go ahead, like, that's a very similar philosophy to one my current PI said. He said that, like, if something was easy and like there's a clear path to it, someone would have done it already. Whereas, like, in research, you're doing something that is never done before, completely different. There's no instructions. Just let things happen and hope for the best. And maybe you might make a discovery that will impact someone.
Who's your PI?
My PI is Dr. Prasani. He works in the chemistry department at UCSD, doing theoretical chemistry. So,
okay.
But, yeah.
It's a good sentence, though. I completely endorse it.
All right. So next question here. This is, I'm going to try to share my screen for this one. It's mainly because I'm going to be trying to draw a little bit. So just a question on how you would explain one of your recent papers. I think you had a paper on HDAC inhibitors, how they tune microRNA in extracellular vesicles. So I was wondering here, like, if you could try to guide me at how to draw this for, say, a high school student, like how to simplify the process so that other people can understand it.
You want to know more about the genesis of this finding? Why did we get into that, or just to explain what we found?
I guess both.
Okay. Because they're connected, so both is fine. So that specific paper stems from the long-standing observation that actually, you know what? I'm going to join with what I told you before. You remember when I told you that the first, let's say, discovery I made, and it took me first to Germany, and then here was the fact that I understood there was an enzymatic activity that was used by MyoD, and that was an acetyltransferase. So that was the way by which MyoD binds the DNA on a promoter of genes, increase the acetylation, and the increased histone acetylation promotes gene expression. But then I asked myself, well, you know, if you in the car, if there is the gas, the accelerator, you also need a brake. So I was guessing if there is that type of situation, good point, you want a brake. So you want to control. And that's what I did, and we found that there was an enzymatic activity that was opposing to the histone acetyltransferases, and that was the histone deacetylase. So the acetyltransferase catalyzed the transfer of an acetyl group. You're a chemist, so you understand that. An acetyl group, the lysine of the histones, and this is called acetylation. And what the HDAC do, the histone deacetylase, is to remove the acetyl group from the histones. Very simple. So it's a dual, and it's like accelerator and the brake. So, and the balance of the dual gives the speed, provides the speed of your car, which in this case is the speed of transcription. That's the way the muscle controls the expression of all the transcripts like contractile proteins, metabolic genes, and all this process that are necessary for muscle to respond to whatever homeostatic perturbation. And so when I understood that HDAC were opposing the transcription of muscle genes, I immediately started to search for drugs that could block the activity of the HDAC. So if you want your car to speed more, I thought, you have to prevent your foot on the brake. That was the drug, and the name was HDAC inhibitors. So there were these drugs that there are many of them that block the HDAC. And I started to play with them. And I still remember so well because it was May. It was May 1999. And I do remember because it was the day after Cinco de Mayo. And I was very late the night because I joined a party and then I completely forgot that I needed to add these drugs to my cells. I went back to the lab. It was 4 o'clock in the night. And I said, okay, let me see how are doing the cells that I treated 3 days ago. And I opened the incubator. I took the dish out of the incubator, put under the microscope. And I saw these muscle fibers that I never saw earlier in my life. And I said, okay, here is an effect. So from that time on, I started to study the potential of HDAC inhibitors to promote muscle growth for muscular dystrophy. And in 2006, we published in Nature Medicine that that was the case. A general HDAC inhibitor was able to prevent the disease progression. This moved, into a clinical trial that is still ongoing. Actually, patients are doing pretty well. I'm very proud of it. And then as I say, how does this happen? And I noticed that one of the reasons why that happened is that other than the muscle fibers, the muscle are composed by several types of cells. And every time the muscle, the myofiber breaks, these cells start to promote the regeneration, the repair. So among these cells, there are the break of the muscles. They're called muscle stem cells or satellite cells. You will go into literature and see that they are called satellite cells because they look like satellites. The satellites are small. Those are small cells around the big fibers in a satellite position, let's say. Distinct from the myonuclei because they are bending at the basal lamina. And other than this, there are, let's say, if those are the break of the muscle, there are the workers, those that need to know how to place, how to instruct the break to form the new muscles. And these sort of organizers are called muscle resident cells. The muscle resident cells don't make muscle by themselves, but provide the structures and the signals for the muscle stem cells to become muscle. However, what we discover is that in muscular dystrophy, at a certain point, this potential is exhausted So if you look at the disease progression of these boys, at the very early stage, they're pretty well compensated. So right now, most of the dystrophic boys can still walk, can still compensate the loss of muscle with this compensatory regeneration for a long time, way longer than we used to see at least 20 years ago. And this is because you have this regeneration ongoing. However, at a certain point, it fails. These cells somehow are lost. And what the muscle do is to get into another type of repair. So instead of repairing the muscle with the break of the muscles, the brick of the muscles, which are the satellite cells. So I say, okay, you know, when you have, for instance, if you have a T-shirt, you know, and you want to repair the T-shirt, and there is a hole, and there is no way to sew back the T-shirt, and you don't want to feel cold, you can put a patch, right? Piece of paper or whatever. You want to fill the gap, but it will never be the same gap as the same material. So this is exactly what the muscle do. So once they don't have the cells to rebuild the muscles, they use the cells to fill the gap. And the filling of the gap is made by fibrosis, by collagen deposition, sometimes even by fat deposition. It's a repair, but the trade-off is that you repair a tissue, but not with the contracted tissue. So it's like with the T-shirt, you know, it's not gonna be, if you stretch the T-shirt, repair in that way, it's gonna break more and more and more. And that's exactly what happened with the muscle. Instead of repairing with the contractile myofibers, it gets repaired with this fibrotic fat material. So in the end, this repair, once the contractile units are depleted or are lost, that's when the children start to go in the wheelchair, because there is no contraction, there's no contractile activity. So what we found is that the HDAC inhibitors were preventing these kind of cells that we called coordinator to produce fibrotic material, and were restoring their ability to activate muscle stem cells. And we say, well, this must be something that is dealing with the interaction, the functional interaction. So here's one of these cells by the way, their name is the fibrodipogenic progenitors, like fibroblasts. They're in between the muscles. And every time the muscle gets lost, they say, okay, we got to organize ourselves. Okay, let's call the stem cells. And that's what the HDAC inhibitors are doing. They're reviving the ability of these fibrodipogenic progenitors. Start organizing the party, you know, okay, let's promote the activation of more muscle stem cells. And at the same time, at this point, they don't need to make fibrosis any longer. So this is it's a curative, it's a therapeutic principle of the HDAC inhibitors. But we wanted to dig into it more and say how HDAC inhibitors revive these functional interactions. And it was clearly by secreted factors. So we started to understand how muscle stem cells and these fibrodipogenic progenitors talk. And there were two ways we were considering. By releasing soluble factors like cytokines, growth factors, mitogens, all these kind of, you know that. Or by another tool that is emerging as a very interesting biological process, which is the production of these extracellular vesicles. The interesting point is that these intracellular vesicles contain proteins, ribonucleic acids, and they can transfer genetic material from one cell to another. And when we tested this hypothesis, we found that inhibiting the production of these extracellular vesicles was sufficient to block the pharmacological activity of HDAC inhibitors. So we say, in science, you first trying to understand what is the ultimate requirement. So you block one activity and you show that that activity is required for something. But then in science, you want to show also the other around. So now I want to isolate that activity. So we isolated the extracellular vesicles and we asked, are they able to actually recapitulate the action of HDAC inhibitors without HDAC inhibitors? So the experiment was to treat the cells with HDAC inhibitors, take the exosomes or the extracellular vesicles, and then use them in vivo to cure dystrophic muscles rather than HDAC inhibitors. And that's what we found. So let's say, if you wish, if you will, we can call this therapeutic surrogate or I don't know, whatever therapeutic essence of HDAC inhibitors. So the next step was, okay, what do they do, HDAC inhibitors, to make such therapeutically effective extracellular vesicles? So we started to study the content, and we started to compare the content of normal cells, extracellular vesicles isolated from normal cells, dystrophic cells, treated or not with HDAC inhibitors. And what we found is that the HDAC inhibitors were completely changing the content of these extracellular vesicles in terms of protein, and especially the focus of that paper is microRNA. And what we found is that most of the microRNA that were induced by HDAC inhibitors were targeting molecules of therapeutic interest. For instance, the Notch pathway, which is a pathway that is supposed to activate, at least implicate in the regulation of stem cells, muscle stem cells. So the discovery there is that the HDAC inhibitors can lift, if you will, can modify the content of these extracellular vesicles and achieve a therapeutic potential by incorporating more and more of these therapeutic microRNA. And our next step now is to understand whether we can produce an homogenic, at this point, we're trying to move into a translational medicine. So we're trying to purify these extracellular vesicles, the most homogeneous population we can, which is a challenge because it's a very heterogeneous population. But when you go into a therapeutic tool, you want something that is homogeneous, you know what it is, and what it does. And so we're at this step, and especially we are trying to understand how these extracellular vesicles are incorporated by other cells. If there is a code that tells them where to go, probably got that. So probably there are some proteins expressed in the surface of these extracellular vesicles that impart to them the ability to find their own target, which I think is the most interesting observation in terms of regenerative medicine. And that's where we stand right now.
Wow. So that's a lot to try to digest. All right, so what I plan to do with this is to kind of quote unquote animate it, is to like kind of draw out what you're saying in the simplified form, like the revival of pharmacogenic progenitors, how they can rescue the muscle. I don't know if you were watching me like trying to draw this out. yeah.
I like your
you
approach.
have-
I mean, it's perfect. I would just follow. I was talking, and I was very, I was very happy to see that my words were translated in a good drawing from you. Actually, it's remarkable. I have to say, I'm impressed. I mean, I didn't take for granted that a student could follow me and draw the right picture real time. So I'm really impressed. Congratulations.
Yeah.
You should keep on doing research.
One of the, I guess, pedagogical techniques used in this class is that they have you draw out a lot of the results you do, or a lot of the expected results if you were to design experiments. Which I think for, like, especially for muscular dystrophy stuff, like, it's really hard to understand without seeing a picture. Like, they tell you all the theory, but unless you see a picture, like, I cannot comprehend it. So yeah, all this is like what the muscle cells look like. This all comes from the experience of drawing things that have undergone fibrosis and regeneration and scarring tissue and all that. And histological, a lot of histological things to study. Right. So,
but it's simpler than you think. Well, there's something that you probably already know that I forgot to tell you. Data analysis. So I'm telling you experiments and how they're. I told you about the rationale of the experiments. I told you about how we plan them, but probably the most challenging step right now is the data analysis, because right now we, at least in my lab, we work at the genome-wide level. So we inspect, for whatever thing we do, we inspect the whole genome. To do that, we use deep sequencing. So we're dealing every day with an enormous amount of data that need to be analyzed in a bioinformatic way. So I always advise every student to get a strong background in maths and bioinformatics. They're really required for.
Funny you should say that. It's actually kind of funny you just say that, because I'm actually signed up for a bioinformatics class next quarter, and I'm huge in trying to learn like machine learning and data science, because like I know that's the big thing in the 21st century is all about data.
That's the best investment you can do, because you're going to deal with millions. That's it, with millions of data. And all you want to do is to understand right away how to order the data, how to eliminate false data, and how to, focus with what we call tracks, your data, and make a calculation. Are you still there? I see your internet connection is unstable. Perfect.
I'm still here. Yeah.
So yeah, go for it. Go for it.
And I guess the last question I have for you here is, like, how do you, I guess, improve pedagogy and trying to inspire more students to get into research like this, whether it be in Duchenne or in any other field of research?
Well, there are 3 levels of inspiration. The first is the most general, and is scientific curiosity. So once again, you may see you may apply it to, it's general because you may apply this to everything in life. You see an object you don't understand, and you start to go around and see, what is this object? Why is there? Who is using this? What is the function? And then, after you observe, you may decide to observe the same object under different conditions, right? So let's see how this object behaves under the sun, under the rain, under the snow, change of temperature. Those are the variables. Already at this level, you have experimental variables. You don't know, but you're using variables, the variable in this case is clearly the change of the weather, the temperature. And then there is the, so this is the level of inspiration about scientific curiosity. You're curious about something, you start to postulate hypotheses, and this is one level. It's typically why students enter a lab. And, you know, at this point, they're dealing, you know, the object is gonna take an identity. It's an object of biomedical interest. It's a biological phenomenon. At this point, there are the two further level of inspirations. There will be another one, but I'll take it at the very end. One is the intellectual challenge. I want to know more. The challenge could be done with your brain or with the brain of your colleagues. It works both sides. You really need colleagues. You really need a competition. There is a part of you that, you know, will find rewarding the fact that you understand things earlier than other people, better than other people. And this is a pure intellectual challenge and has to exist in every scientist. If you don't have that, it's a big problem. But this has to be kept in balance in a very, very important equilibrium with other inspiration, which is, I want to solve a problem that is a problem for somebody else, that is destroying the life of a family, or, you know, is affecting the really, you know, the society, I think. But more than this, I would say you start to feel, you start to enter into the brain, the heart of parents, patients, and you really want to help. At this point, your competitors, your colleagues need to become your friend. So you have to always keep in equilibrium your normal physiological tendency to be the best, and the acknowledgement that other people can be better than you, and that everybody is going to provide a complementary information. So a good scientist should recognize a good scientist, good science, and put together the pieces. And sometimes you have to swallow your pride. Somebody arrived before me. There's nothing wrong. It was better than me. Next time I will be better than him. But what is important is that me and him are going to move the field with discoveries. So when students, after the first level of interest, join my lab, I always try to motivate them both with the level of inspiration too. You can understand better than anybody else. And the level of inspiration number 3. You may want to really help people that have been clearly unlucky, you know, less lucky than you. And that's, I believe, I don't know, you should ask people in my lab, but in my opinion, because I see that when people leave my lab, they continue to work on muscular dystrophy. This is the strongest, the strongest motivation is the motivation that remains all your life. You know, I try as much as I can to expose these people and understand that once you start to work on a certain disease like this, there are patients that rely on your work. They trust you, they believe you. And if you start to, you know, if you leave that research or you start to get corrupted by money and, you know, your research is not going to be genuine any longer, you are betraying them. So I really work a lot on what, if you will, can call non-scientific motivations. because I think there is a part of us, the non-scientific part, that should be always there. And trust me, it's the part that stays when science fails. So it helps a lot. The fourth point would be the academic career, the professional achievement. There is no doubt that this exists, and I do always my best to form good scientists, honest, that are committed to research, but they have the tool to move on with their academic or industrial career. So it's very important that when they work in my lab, they perceive the fact that their career is moving. There's nothing worse than feeling the same place, the same position for years and nothing happens. So those are the four major things. As you can see, they're very interconnected. And then there may be other small details, but I would say those are the four more important things in terms of inspiration.
Okay. So that's something I look forward to in the future. And being just a junior,
but don't forget something, Steve.
Yeah.
There is one point that's not up to me, but science is fun. It's fun, gotta be fun. It's painful sometimes because you may need to do experiments in the night, a lot of pressure, but it's fun because I'll tell you something. Give me one job where you can wake up in the morning, think about something, and translate your thoughts, your idea into an experiment and verify the same day. Am I true or not? Only science. This intellectual freedom, there's nobody, once you're in a lab, it's a matter of your creativity, your brain. You think and you verify. Other jobs, they don't provide this chance. Maybe you're well paid, but you will never have this beautiful opportunity to challenge your brain and verify. It's great. It's what I call intellectual freedom. And I'm so happy that I do that. For me, sometimes even, like to surf, start to paddle out, take a wave, and then while I'm waiting the wave, I say, oh, this could be a good idea. All the problem is that I may forget the idea. But if I don't, I go to the lab and I translate immediately into an experiment. And it's beautiful.
Well, that's also like one of the reasons why I'm hesitant to just focus on just med school, because I know it's just study, and then you eventually follow a procedure. And yeah, it's well established and you just work hard and you will get paid a lot. But as a researcher, it's you're all about inventing something new. And I think that is a lot more fun than just reading from a textbook or going through information that hasn't really been established for decades now.
No doubt about that.
Well, that's pretty much all the questions I have, unless someone asks me something.
Well, I mean, at this point, I'm available. One thing that I didn't tell you is that I really care about selecting and trying to educate as much as I can bright students. You really look like one of them. So happy to further help you. If you need to write an essay or whatever you need to write, no problem, send to me, ask me material, send to me whatever you wrote, and I can edit and review. I'll be more than happy. But more than this, if by any chance happen to be interested in this and you want to stop by my lab, you're more than welcome. Let me know, I'll let you in, I'll show you what we do. And if there is any chance, somebody like you with your motivation and your brain can spend some time in my lab, it's open for you.
So I hope you enjoyed that interview slash animation slash podcast with Dr. Puri here on his journey to becoming a researcher for Duchenne muscular dystrophy. And hopefully you became inspired to actually become a researcher on top of becoming a doctor, if that's within your goals, or understand and appreciate what it takes to become a researcher slash scientist. And here I have a couple of review questions here to try to take out some of the important messages from the interview itself. So the first question I have for you is, why transition from a doctor to a researcher? Or why did Dr. Puri transition from a doctor to a researcher? Pause the video and answer that. So Dr. Puri transitioned from a doctor to a researcher. She initially studied internal medicine and got a medical degree first, and he transitioned to getting a PhD as well. So initially here, he became a doctor because he wanted to work in the ER. He wanted to work in the ER and he wanted to throw on the excitement here. But what wasn't really appealing of becoming a doctor was that he would just read books and procedures and just perform them. And while there's nothing inherently wrong with that, you could just read a bunch of books, read a bunch of medical textbooks, read a bunch of literature, and apply such procedures. It wasn't really in his passion or interest here. What he was much more passionate about was discovering something completely different, something new here, and innovating and finding new ways to cure the uncurable, or things that have yet to be cured. And he realized that if he was a doctor, that he would be stuck performing the same techniques that have been discovered already. He wants to do instead the discovering. He wants to discover new methods here, new techniques that can potentially help others that we don't have ways to cure for yet. And that's the transition from becoming a doctor to becoming a researcher for Dr. Puri. Next question here. Why UC San Diego? Pause the video and answer now. So UC San Diego is known for a few things, more than a few things really, but one of its most prominent geographical characteristics is that it's right next to the beach. And in fact, here, Dr. Puri here wanted to go to the beach. He did his postdoc at UC San Diego because of that. And he wanted to go to the ocean, he wants to go to the waves, he wants to go surfing. So that is why UC San Diego. We are right next to the beach. And if you do visit UC San Diego, depending on the western side of the campus, so the part that's closer to the ocean, the beach should either be, say, a 10 to 15-minute bus ride away, or a 5-minute walk, depending on how far you want to go. So that's why UC San Diego. The next part's a little more technical here. How do HDAC inhibitors work? And what are their relevance in Dr. Puri's research? Pause the video and try to answer on your own. So this one's a little bit trickier, and it's the scholarly or academic part of the interview. But the important part behind this is that it shows that it's not just about trying to find this new medicine or this new cure, but it's also about making sure that the medicine actually works and is maintained over time here. And that's the emerging field of translational medicine, or emerging concept in translational medicine. So in the interview, I drew cells here that had an acetyl group, an acetylene group attached to it. And what HDAC does is that it removes that acetyl group, or acetylene group, sorry, from the cell. What HDAC inhibitor does, as the name implies, is that it slows down the process here. It's an inhibitor. And what Dr. Puri mentioned here is that ratio between cells that are acetylene versus cells that are not, so cells that have undergone deacetylation. If we have a higher ratio between the two, then the transcription speed goes up. We use the gas in the brake and the speedometer analogy to help us understand how the microRNA that Dr. Puri inserted, or his lab inserted into the cells here, actually gets expressed here. So we have transcription. Now we take our microRNA, and we've seen this in class here. MicroRNA, you put into the cell and we can produce dystrophin here. But what we haven't seen here is the fact that this microRNA needs to be transcribed and needs to be translated. It needs to create all these proteins here. Really, it needs to create dystrophin here. While we consider the method, we haven't, in terms of getting microRNA into the cell, we haven't considered the speed here and maintaining that over time. So by having more of these H-type inhibitors here, you put more effort on the gas, leading to more transcription of the microRNA. That increases the amount of dystrophin that the cells can produce. That in turn reduces fibrosis and inflammation. An analogy to this would be in, say, nanomedicine or nanoengineering, in which nanomedicine here, we're considering, say, the pharmacokinetics and the biodistribution. Or in other words, how fast does the nanomedicine get to this particular target, and how long of an effect it will have here. The same idea happens with H-type inhibitors, in which we want the H-type inhibitors to have a lasting effect on the muscle cells here, and making sure that the microRNA is actually being expressed here in the form of dystrophin. So we want to have that right ratio here, because too much dystrophin is bad, really too much of anything is bad, but too little means that the treatment is ineffective here. So we're looking for that balance, but increasing the amount of dystrophin here can help counter the muscular dystrophy. The next point is a little more subtle, or a little bit harder to find in the interview. What is the next innovative field that lies at the intersection of statistics, biology, and computer science? Pause the video and try to answer on your own. So you may recall the diagram that I drew in the beginning. If I can draw the diagram again, in which I put statistics, biology, and computer science. And in the middle, at the intersection of all three, is bioinformatics. Bioinformatics here. You may have heard of something called the Human Genome Project, an ambitious project that tries to map out the entire human genome. And while completed, or just continuous innovations or adjustments to the Human Genome Project, with bioinformatics, we can introduce new computer science algorithms. We can use statistics to apply new tools into biology that helps us solve problems that we wouldn't be able to solve by hand. Things like machine learning, there's algorithms, estimations, predictions. This is where statistics and computer science come into play here to help us understand certain biological phenomena that we would never understand or have a hard time finding out until much, much later. So an analogy to this here will be in the field, and this is more close to my heart, is in computational chemistry, in which we use machine learning and other data science methods to be able to predict the behavior of certain molecules here. And in fact, we can use computational chemistry here to predict the structures of various proteins in the realm of chemical informatics to be able to predict which organic chemicals we can use in drug design from all these chemical databases that hold a myriad of information. And we can apply those methods here into the realm of computational biology. And not just in human physiology here. We can use it to predict how animals move around in an ecosystem here. We use computational biology, more so in physiology here, to predict, say, how certain nanomedicine here goes throughout the body in terms of pharmacokinetics and biodistribution. We can use bioinformatics here to figure out which proteins are particularly useful for us in terms of drug development and design here. And while we're not yet at the point in which we could, say, model the human body accurately 100% of the time with just a computer or simulations, and what I mean by that is on a microscopic level, we're taking things step by step here to try to understand how we can use science, chemistry, law, statistics, computer science, and combine them all together into the realm of biology to try to really understand the human body from a more computational perspective. Finally, and this I think is the biggest takeaway from the entire interview, is what are the 3 to 4 steps of inspiration necessary to become, quote unquote, a successful scientist? Pause the video and try to answer this on your own. So for most of us, it could just be 3 steps, or for some of us, it could be a 4th step here. The first step is establishing that curiosity. You need to be curious and passionate in your field in order to be able to come up with new ideas. Because if you lack that passion, if you lack that creativity, then your best bet in becoming quote unquote successful in that field is just reading a book and applying a procedure. And while there's nothing inherently wrong with that here, to become a researcher involves coming up with new ideas, things that are novel and change the research landscape. The next step here is to be able to work with others. Not just because teamwork is better than solo work, sometimes solo work is better, but also it provides extra motivation here to try to learn more and try to push on ahead. Dr. Peary mentioned that friendly competition is a great thing to have because it forces us to become much more motivated to try to get ahead here. But at the same time, we also want to rely on our fellow scientists for their expertise, as no one scientist is great at everything here. In fact, becoming an expert in everything is never the goal, but rather becoming an expert in being able to lean on others for their expertise. Because there will always be someone that's better in one particular field than us. So being able to lean on other scientists for extra information to help solve a problem that without current knowledge we wouldn't be able to solve here is an essential step to coming up with something new and innovative here, but also it works, hopefully. The third step here is really trying to be able to help the community here. Being able to take things one step at a time and give them back to others here. So while we do, as researchers, gain a lot of training, we want to try to give back to the community here. We still want to help others because our research projects here are only good if others can benefit from that here. And while we could discover something new, if no one knows about it, it's like we haven't discovered anything at all. The final point, and this isn't an essential prereq, but it's something that that people desire, it's something that people want, is having extensive training in graduate school an MD or PhD here, but also more importantly, to continue rising and to continue growing here, because we stay in one place. Going back to Dr. Pierce's analogy and how on that missing object, if we stay in one place, we're not able to find a new perspective. And without finding a new perspective, that missing object will remain missing. But by walking around and taking a new perspective on an issue, and by gaining new experiences, we're able to develop something innovative, something new, to potentially help us find a cure for DMD. And that is the main takeaway of this interview here, is to find a way to become innovative here, because the cure is out there. It's just a matter of finding it. So I hope you enjoyed this interview and are inspired to, if not in DMD, to become a researcher in the future, or at least think like one, because these are the people we need in order to create new solutions that can help make the world a better place. Thank you for your time, and I will see you next time in another one of these interviews, if they happen in the future.